Thank you, Martin. Just a minute. There's a reason for this. I have a 15-year-old daughter at home. She still goes to school. And I'm constantly confronted with the question, Dad, when you go away, what do you do really? Because she cannot, in her wildest dreams, imagine that anybody would sit down voluntarily and listen to me for more than 30 seconds. So, evidence. Today we'll be talking about origin of life at a hydrothermal vent. Okay, important, water. Everything is underwater. So we're going to be making an argument that life arose from hydrogen and CO2 in an aqueous environment catalyzed by transition metals. I feel sometimes a need to justify my research, and so for Wetsus it's easy. Everybody needs water. We heard all about that yesterday. We don't need to get into that. But it's interesting also if you think about origins. Every society on Earth has a story for where everything began, including scientists. And I don't know why that is. That would actually be interesting to investigate from a philosophical standpoint, but it seems to be part of our human condition to want to know where we come from. That's something very natural. And scientists also have their narratives. Sometimes theirs are the most imaginative. And so that's a justification. Why do I investigate origins? It's because I want to know, and I'm not the only one who wants to know. We all want to know how things began, how it was possible that on a young planet with cooling magma, water, nitrogen atmosphere, and CO2, life could have arisen and given rise to scientists at meetings. So that seems unimaginable, and the goal of my talk is in the next 45 minutes to make that more imaginable. And it doesn't make any difference whether or not the hypothesis that we will be hearing today is true or not, because I already have a permanent position. and the role of investigations like this are to structure the problem so we can think about it and work on it in the future. So, as a biologist, I see that life is a chemical reaction. If you don't believe me, try to hold your breath until the end of my talk, and then we will have to agree. Life is a chemical reaction. It's an energy-releasing chemical reaction, and it's not just the reorganization of pre-existing components. It's a chemical reaction that organizes material in the process of releasing energy, and the question that I want to get in today is, from what chemical reaction did it start? So, if we're talking about evolution, origins, everything in evolution starts here with Darwin's first picture and showing his two principles. Natural variation, the individuals at each population are not identical. He observed this from going to pigeon breeding shows and dog breeding shows. And then his big idea, natural selection. Some of them are more fit than others to make it into the next generation. Using this, we can explain the entire diversity of the known living world, except there's a problem with origins right down here. And that is, it works much better, Darwin's theory works much better if you already have something that is alive. And in the transition from things that are not alive to alive, we have to work with different principles. This is Haeckel's tree. Haeckel was great at drawing the relationships of all things big and small, known to the then world of biologists. And his famous tree in 1866 shows a monophyletic tree of organisms. But when it comes to origins, he's also kind of cheating because there are no roots here. He's mentioned something about reactions of CO2 and ammonia in inorganic salts, but never gets very explicit. So we can explain, this is Darwin's evolutionary principle extrapolated to the then known living world. He's got the prokaryotes down here. But it doesn't explain origins. Also, it's interesting to note that we always think of this as the culminating work of a picture of a man's life's work. That's not true. When Haeckel drew this, he was 32. When he published it, he was 32. So He was quite a talent. This is Konstantin Mariskowski. This is a different view of origins because if you look down here, right, there's something going on. Mariskowski had a different view of life's evolution in that he had symbiosis built into it. You'll notice in Haeckel's view, everything is diverging. Mariskowski worked in physiology, comparative physiology, and he noticed that the chloroplasts of higher plants must have been cyanobacteria. This is 1910, this is a very prescient observation. And then down here at the origin of life, he had these biococci, some reactions going on down here. And just from comparative physiology, which is something that I still do today, he inferred the following. So, I can't, my glasses. All right, so a minimal size, okay? He inferred that the first organisms had to be very small, okay? And maybe invisible for the microscope, that they had to lack organization, all right? This would mean in today's language they were prokaryotes, not eukaryotes, so very simply organized. They had the ability to withstand very high temperatures. This is directly from his 1910 paper, okay? So that's thermophilic origins, or a hot start. That's a very modern concept. They had the ability to live without oxygen, okay? He didn't have the origin of oxygen pinned to cyanobacteria and a later addition, but he knew that the first cells had to be anaerobes. Okay, and here I know this one by heart, the ability to synthesize proteins and carbohydrates, the latter without the help of chlorophyll from inorganic substances. Now, translated to modern terms, that's chemolithic autotrophic origins, anaerobic and thermophilic. That's a very modern concept, and we're still working on those lines today. So anybody who says this is a new idea, it's 1910, the old literature is vast. And this is also resistant to high salts and poisons. So that's kind of an interesting idea that was very popular at the time. It became unpopular around 1923 by these two gentlemen here. They published independently of one another. This is Oparin working in Moscow and Haldane working in London, the idea of organic soup. And so I can't see my audience very well. Many of you will have heard of something like organic soup or Ursuppe. How many of you have heard of organic soup or Ursuppe? Aha, everybody. This is very interesting. It's a very widespread concept. I'll show you in a minute. It won't work, okay? But it's a very widespread concept. All right, so how does organic soup theory go? All right, so Haldane's version is much shorter and much easier to read, and it goes something like this. On the early Earth, there was no life, but there was CO2 in the oceans, and there was UV light, and UV light, it was known then, would react with CO2 in the presence of inorganic salts and in the presence of water to give reduced organic compounds. These could accumulate because there was nothing there to eat up all those organic compounds, and they would accumulate and accumulate, get more complex and more complex, make up something on the order of macromolecules, then virus. Haldane had virus as an intermediate from the simple molecules to life, and then because it's only a 10-page paper, he kind of leaves out all the details, so the waves get high, and there's lots of wind and thunder and lightning, and then out calls the first life, and that first life was heterotrophic, okay, in contrast to the autotrophic origins of Mariskowski. It was heterotrophic, and it would eat up all the soup, and that provided the selective pressure for the origin of photosynthesis, and then the sky's been blue ever since. Okay, so we lost autotrophic origins, and we lost chemolithic autotrophy, all right, in that. Now, okay, if you don't remember anything else from today's talk, let's remember this. Under no circumstances can this organic soup theory work. Let's do the experiment. Stop laughing. Under no circumstances. Let's go to the grocery store, and we get a can of chicken soup. We get a can of goulash soup. We go to the laboratory, take 3 frogs and 4 carrots. We homogenize them, okay, but in a sterile manner so that we have all the molecules that we need, fresh molecules for life. Put that in a can, sterile. And then we take water as the control and put those 4 cans at any temperature for 4 billion years, and we wait for new life to arise de novo. Is it going to work? Those of you who are laughing are paying attention, okay? It's not going to work. The more difficult question is, why not? And it's because the carbon and the nitrogen and the oxygen and the hydrogen in those cans already has reacted. It has almost no potential to react further. Life is a chemical reaction. The molecules of life, amino acids, bases, lipids, are low energy, okay? They're stable and they're at equilibrium for the most part, and therefore they have no potential to react further. And that's why organic soup theory won't work. Life is a chemical reaction. Can anybody tell me what these two gentlemen have in common? A mustache. Okay, I heard it from the front row. That had to be a professor. It's always a professor who says they had a mustache. Yes, they have a mustache. Can anybody say what else they have in common? Sorry? Jackets and ties. Okay, also from the front row. No, they were communists. They were communists. Now, I forgot to tell you that I come from Texas, right, originally. I don't come from Germany. And so I know what you're thinking now. Okay, he comes from Texas. He's got a problem with the communists. This all sounds really familiar. But no, it's dialectic materialism, because this is just after the October Revolution, and in Marxism-Leninism, you had to have a rational explanation for the origin of everything, from the working of society to the needs of individuals to the origin of life. And actually, this organic soup became party program. So let's move on from there and get to the topic. Okay, we're going to be talking about hydrothermal vents. And there are a lot of people to be mentioned here. We should mention John Barris in particular, who in 1985 was the first to really make the strong argument that these newly discovered hydrothermal vents that they called black smokers, discovered in 1978, were important for the origin of life. And Mike, he was working on a different kind of vent that was not a black smoker, but he was working on it from a geochemical perspective, because as he thought that these low temperature vents with very alkaline and hydrogen rich solution were important for the deposition of what are called giant ore bodies. These are the things where we get our cadmium and heavy metals, lead, tin, also a lot of gold and silver. And there are 14 of them around the world. And he said, we propose that life arose from growing aggregates of iron sulfide bubbles containing alkaline and highly reduced hydrothermal solution. Those bubbles were produced at hydrostatically at a sulfidic submarine site sited some distance from a spreading center 4 billion years ago. Well, the type of vent that he was talking about here wasn't discovered until Deb City and her colleagues discovered a place called Lost City, which you can Google. And Lost City is exactly that kind of hydrothermal vent. It contains cool water, about 70 to 90 degrees. It's very rich in molecular hydrogen, which is a source of electrons and energy if you put it together with the right electron acceptor. And they also form bubble-like microcompartments. Well, what is the rock that Mike was so excited about? It's this. These are called botryoids. Here you can see the scale. All right, we're looking at the surface of a submarine hydrothermal vent. This chimney over here is hollow in the middle. It's water coming out, and it's about as big around as this laser pointer. And you can see these little bubbles over here. These are called botryoids, or grape rocks, and they're inflated, okay? They are hollow on the inside, inflated by the hydrostatic pressure of iron 2 rich water, in this case meeting sulfide rich water at the bottom of the ocean, giving iron sulfide precipitates that fall out, that precipitate colloidally and produce these wall-like structures, okay? These are not made by organisms, these are made by this precipitation. Now, they're made of iron sulfide, or transition metal sulfides, and they're going to play a very important role in today's talk and in today's theory, because they will fulfill two very important functions. Number one, they provide a system of compartmentation. Regardless of what theory you assume for the origin of life, you have to have the compounds concentrated around their site of synthesis, because if they diffuse off into the ocean or are washed out into a river, they're lost in the process of further molecular organization towards higher complexity. So there has to be some kind of compartmentation. These naturally forming microcompartments will solve that problem quite nicely. And number two, for the chemists in the audience, I don't have to say this, but for the others I will, they're made of transition metals. Now these transition metals, if this is an iron atom, or could be molybdenum or nickel or cobalt, what they have are something that nitrogen and carbon and oxygen don't have. They have d electrons. They have a d electron shell, and these d electrons have unusual orbitals that can hybridize in their geometry and in their properties such that they can form metastable bonds with carbon and nitrogen up here, so that the carbon and nitrogen can react, and then the d electron orbital is pulled back in, and then the carbon or nitrogen can leave reacted and changed, whereas the metal is unchanged. That's the property of a catalyst. It leaves the reaction unchanged. And the important thing is that even though today chemists in industry use coordination chemistry to modify, they can modify the properties of these d orbitals to get specificity and particular attributes of these catalysts. But the property of catalysis is a property of the element itself, okay? The elements themselves. We don't have to do anything to them to make them catalytic. They have that natural catalytic ability, and that's important. So we've got naturally forming walls at a hydrothermal vent made out of catalytic material, and there's hydrogen at that vent, and it's going to be meeting CO2. That's a good prerequisite for something to happen. All right, so we're not talking about black smokers like this, because the black smokers sit right on top of the spreading zones. The water coming out of the black smokers is about 400 degrees. That's too hot for life. Also thermodynamically not good for reducing CO2 to reduced organic compounds. We're talking about lost city type vents. These are much cooler, very hydrogen rich. They also have the black smokers sit on the spreading zones. They have a lifespan on the order of about 10 years to 50 years, and Lost City is 30,000 to 100,000 years old. It's continuous because it's not on top of the spreading zone. This here is calcium magnesium carbonate. It's not iron sulfur, but it does form microcompartments. These are, if you cut off a piece of Lost City and then make an electron micrograph of it, you can see the scale here, 500 microns and 60 meters high. That's a lot of surface area. And then we use our imagination, okay, just to get to the point. So about 2002, Mike Russell and I got together, and I made this figure, and this sort of puts the whole thing together. We have hydrogen down here, CO2 here, and where the hydrogen comes from, we'll get to in a minute. Where the CO2 comes from is actually quite important. Okay, we know that shortly after the Earth formed, there was a large impact by a planet called Theia. It was about the size of Mars. It hit the Earth at a little bit oblique angle, and that gave the Earth its 23-degree tilt, and it melted the Earth and threw a bunch of magma out into space, from which the moon formed. And the moon is now at a 5-degree angle around that. And in the process of melting the Earth, converting the Earth into molten rock, and some of it was vaporized, that did two important things. Number one, all the water that was on the early Earth, water, was transferred to the gas phase, because water is not happy in 1200 degree or 2500 degree magma. And it also took all the carbon on the early Earth, converted it to CO2, and put it into the atmosphere. Okay? So that takes, on the early Earth, we've got all the CO2 out of the, basically out of the mantle, out of the primordial crust in the atmosphere, interfacing with hydrogen from hydrothermal vents, and how that's made we'll get to in a minute. All right, so this gives us in microbial ecology, this is called in physiology, a redox couple. Rolf Tauer explained it to me once very directly. He said, in the reaction of hydrogen with CO2, the equilibrium lies on the side of reduced carbon compounds. That means we don't have to do anything with these two. They want to form methane, acetate, and methyl groups all by themselves, because the reduced carbon compounds are more stable than the two input compounds. So we get thioesters, amino acids, all in this theoretical hydrothermal vent containing these catalytically walled compartments, higher steps to higher complexity, you can imagine it. So I'll try this story. I'll try the short version. So when this figure appeared in the Philosophical Transactions of the Royal Society of London, the world's oldest continuously published journal. There was a lot of press and reporters calling up all the time, and also the Catholic Academy in Stuttgart called up. Okay, so I'm sitting in my office, the telephone rings, I answer the phone. Herr Martin, yes, this is Schmidt from the Catholic Academy in Stuttgart. We'd like to talk to you. What about? It's about your recent paper. On the origin of life? Yes. Am I in trouble? No. We just want to talk to you. And so they asked me, say, we'd like you to come down to Stuttgart and give us what we call an impulse. And that is, you stand up on stage and have to talk for 2 hours telling all the details you know, and we get to ask as many questions as possible so that we can be informed about this. I said, this doesn't have anything to do with the Inquisition, does it? And said, no, we just want to know. And it's actually true, because this was shortly after Johannes Paul II opened up the church to the concept of evolution, said we have to talk about it, right? We don't have to accept it, but we have to talk about it. So I did that, and I went down, and there were only, in contrast to today, there were only 12 people in the audience. There were 3 priests, 3 scientific theologians, 3 philosophers who wanted me to define everything, that was very annoying, and 3 physicists. Now, can you tell me why origin of life always has physicists involved? It's a biological problem, not a physical. All right, so I'm doing this talk and I'm showing this slide, and then one of the priests asked me here, says, Dr. Martin, so I can imagine how we can get from H2 and CO2 to amino acids and bases, and I can also imagine once we have things like cofactors that provide organic catalysis within these systems to get into what are called autocatalytic networks, then I can see how we can get to RNA and maybe information and genes and stuff like that. But the transition from amino acids to cofactors, that seems very difficult. Wouldn't it help if you used a little bit of God? Okay, and the answer is, oh yes, and the more God you use, the better it works, okay? But we're not allowed to do that as scientists. When we don't have an answer, we can't say it was a miracle, all right? That would shorten our, that would make things, that would shorten our activity dramatically. We just have to say we don't know and then push on. All right, so that's that. An important part of this, in this theory, the last universal common ancestor is not free living, rather it's confined to the compartments within which it arose. And the escape to the free living lifestyle comes later. And it's constantly supported by this flow of energy of H2 and CO2. The iron sulfur clusters of modern proteins that we see in the proteins of in particular chemolith autotrophs, but also in our mitochondria. These are not inventions of the biological world, rather they're just incorporations of the mineral structures from within which life arose, because the structure of the iron sulfur minerals is not fundamentally different from the structure of the iron sulfur clusters in modern proteins. Okay, so then the last universal common ancestor differentiates, and eukaryotes have nothing to do with the origin of life in this theory. It's Archaea and Bacteria, the 2 kinds of prokaryotes. And one of the issues that we deal with here is that compartmentation comes late. That would help to explain why Archaea and Bacteria have the same universal metabolism, okay, for the amino acids and bases, and mostly the same cofactors, and they have the same genetic code, but the isoprene ethers of the membranes in the Archaea have nothing to do with the fatty acid esters and the lipids of the bacteria. Also, the stereochemistry at glycerol phosphate is different, and the peptidoglycan layer of bacteria is unrelated to the S layer of Archaea. So for any other pathway, we would say that was independently arisen. All right, so that's the theory. And what were these first 2 kinds of cells? Well, I think these were acetogens and methanogens, and we'll get to more of that in a minute. But the main thing down here is that this independent origin. All right, so we've got independent origins of the lipid and cell wall biosynthetic pathways. There are actually a lot of things that separate bacteria and archaea, also 30 ribosomal proteins. So how much can we actually trace to the universal common ancestor? And this is where we did some no, wait a second, stop. Okay, I'm getting ahead of myself. Where does the hydrogen come from? Where does the hydrogen come from that runs this reaction? It comes from a process called serpentinization. This is actually an important word in today's talk, and it's the process that brings forth this hydrogen. And so what happens is that the marine crust is made up of iron magnesium silicate, and this is iron 2 in the 2 oxidation state, and that reacts with water that is seeping down into the cracks of the Earth's crust from the oceans, and then reacting with that iron 2, and in the process of that reaction, the silicates undergo some reactions, but the main thing that happens is that iron 2 reduces water to give you hydrogen, and iron 3 remains as magnetite in the rocks. So basically what happens is that water is reduced to hydrogen in the bowels of the earth, and this hydrogen emerges through these hydrothermal vents, and some of the vents today have up to 50 millimolar hydrogen coming out. There's a lot coming out of these hydrothermal vents, and furthermore, it's been doing so since there was water on Earth, because this itself is an exergonic reaction. It releases energy. Okay, under the conditions of the high pressure and high temperature conditions of the deep Earth, hydrogen is constantly produced from geochemical systems. So as soon as there was liquid water on Earth, this reaction started. And the sooner it started, at the early phases, there was probably more hydrogen being produced. All right, so that's important. Serpentinite is the rock that's made. That's this compound here. This is serpentinite, and it has this green color, and maybe you see it in banks or hotels, right? So this familiar rock. Where the veins are here, that's where water was flowing through at 1 to 3 kilometers, maybe 4 to 5 kilometers below the surface of the Earth, and that's where the reaction was taking place that made hydrogen. So the next time you see that rock, that's where life arose. As a suggestion. All right, so what are the processes here? Acetogenesis and methanogenesis, all right? The reaction of hydrogen and CO2. Under hydrothermal conditions, the reactions that acetogens, these are bacteria, use to gain energy and carbon are this right here. And so we said the reaction the equilibrium lies on the side of reduced carbon compounds. That means that this delta G value here is negative, the change in free energy for the reaction. It's that energy change that they use to pump protons and then make ATP, but also satisfy their carbon needs from CO2 and H2. Methanogen's much the same way, slightly more exergonic because the end product is more reduced. But even to the level of the energy-rich thioester, that is the central compound in these pathways, acetyl-CoA, right? It's an energy-rich compound. It's a thioester. Even there, the reaction is exergonic. So basically what we're getting here is organisms that are getting a free lunch that they're paid to eat. The chemistry of these two reactions is interesting. All right, you can see formal methanol, methylene, and methyl. Okay, so we're reducing a CO2 to a methyl group in both the acetogens and the methanogens. But it's interesting to note that the enzymes of these two pathways are not related. Also, the cofactors are not related. This is actually also another one of these things that arose independently en route from LUCA to ESCAPE. But this central reaction here, carbon monoxide dehydrogenase, that takes that methyl group and condenses it with carbon monoxide that it synthesizes from CO2 and H2, this is conserved, and that will be important in just a minute. The interesting thing is that Lost City not only has 10 millimolar hydrogen, it has 1 millimolar methane of geological origin. And so what we're saying is that this abiotic synthesis pathway from H2 and CO2 to methane, which is exergonic, which is why this takes place at Lost City, is the precursor of this chemistry here. And some people say, Dr. Martin, how can you propose that this pathway is ancient even though the two pathways are not related? And the answer is, the chemistry is older than the genes that catalyze the reactions. And that means that you had a pre-existing chemistry, and then when genes finally, and enzymes came along to catalyze these reactions, all they did was add rate and specificity to pre-existing reactions. And that's because enzymes do not perform feats of magic. They just accelerate reactions that tend to take place anyway. So now we're going to change course a lot because I've been wondering about this. How can we, if this theory is correct, then we have to be able to get some of this chemistry to work in the laboratory using transition metals. And people have had a lot of difficulty doing that, either with titanium oxide without catalyst or using iron sulfur. But the problem with iron sulfur minerals is that it's always a transition from Fe2 to Fe3, which is a 1 electron transfer. And in biology, CO2 reduction is always a 2 electron reaction. It's a hydride being transferred. It's a 2 electron reaction, which is different from a 1 electron reaction. And so I was saying, well, how might this have happened on the early Earth? And then I was looking at the the reaction mechanism of CODH, this central enzyme here that is shared by the two pathways that actually makes the acetyl group. And I said, well, look at this here. In making carbon monoxide, we've got nickel 0, formerly at the oxidation state of nickel 0, because the 2 electrons that end up on CO2 to make carbon monoxide, they're replaced over here. And so native metals, and I started thinking about native metals. And if you look into literature, these hydrothermal vents, they contain native metals. Awaruite is the name. Awaruite is Ni3Fe, okay? It is made from the divalent metal ions through reduction with molecular hydrogen in the hydrothermal vents. That's where it comes from. It's a natural, these are actually from meteorites here, but the nanoparticular awaruite made in vents is interesting. And so we got together with Harlan Tusuz at the Max Planck Institute in Mulheim, who can make nanoparticular awaruite for us. And we started getting some CO, and then we started getting some methane together with Chris Greenwell. And then a friend in Strasbourg started having a lot of success using iron 0, which is different from this because the iron 0 actually reacts to become iron 2. It's the reductant. So then we started using CO2 and H2 in the presence of awaruite. And then we got formate, acetate, ethanol, and pyruvate overnight at 80 degrees to 100 degrees, all right? And what I'm going to show you in a minute is that that is the backbone of carbon and energy metabolism in acetogens and methanogens. It unfolds overnight using a simple awaruite catalyst, one catalyst, to give us these products. And this is the work of Martina Preiner. Okay, I can't say that enough. Martina did fantastic work, and she's now in Utrecht. She's moved to the geology group in Utrecht. They're also doing some work on Texel on nanoparticles and hydrothermal vents. And I'm so proud of the work that she's done. She really got this going all by herself, and this is the reactor that we've been using. So if we look, and this is just to show that if we do the X-ray diffraction of the awaruite before and after the reaction, it's unchanged. There's a little bit of oxidation down here, maybe a carbonate, but it's really a catalyst, okay? So it's not reacting, it's a catalyst, which is exactly what we want. This is the pathway that microbes use, okay, the acetyl-CoA pathway. CO2, formate, formal, methylenyl, methylene, methyl, okay? Acetyl, carbon monoxide, make acetate, methane, acetate, and pyruvate. That's the biological pathway. And these things here are nitrogen atoms. This here is either a nickel or a sulfur atom, all right? So in the cofactors. These are the products that we get in our reaction. With awaruite, we get exactly formate, methanol, methane, acetate, pyruvate. Nothing else. It's exactly microbial metabolism, and these are physiologically relevant amounts. We get like 10 micromolar pyruvate, and the physiological concentrate of pyruvate in exponentially growing E. coli is about 50 micromolar. It's actually pretty amazing. There's something very natural about microbial metabolism. All right, so that brings us back to this. Hydrogen, CO2, so we've got our stock, feedstock getting to pyruvate, and we've got some independent reactions down here. What else is going on? How much of this is independent? How many of these reactions can we trace to LUCA? We did some bioinformatics not too long ago looking for enzymes that in all genomes, 5,000, 6,000 genomes that we could trace to the last universal common ancestor, LUCA. If you just look at things that are present in all genomes, it's only 30. These are ribosomal proteins, we know that. Present in both domains, bacteria and archaea, that's about 11,000, but that could be either presence in LUCA or lateral gene transfer. And so to discriminate, we just looked for genes that are present in both domains in at least 2 groups, and those preserve monophyly. That is, we're taking out the lateral gene transfer. That uncovers 355 genes that were very exciting because it was not only ribosomal proteins and translation and stuff we knew, there was energy metabolism, cofactor biosynthesis, amino acid biosynthesis. What did we get? It's this picture here of LUCA, okay? So from genomes, we get exactly the same thing. Basically, the acetyl-CoA pathway working off of geochemically synthesized methyl groups because the methyl pathways are not conserved. The CODH reaction giving rise to thioesters, acyl phosphates as energy source, half alive, okay, there's not enough genes here to have a free living cell, and it's living actually probably off this naturally formed ion gradient. The ancient oceans were about pH 6, and these hydrothermal vent effluents are about pH 9 because of the serpentinization process. That's an ion gradient that they would be able to harness using a simple protein like an HPase. All right. How much further can we go with this? We looked at the basics of, getting to these cofactors. The metabolism of LUCA here, it's only 355 proteins, but those proteins require all 18 cofactors that we know. And so we said, well, what is the metabolic challenge to get to cofactors and amino acids, the building blocks, not the polymers, but the monomers from which life is made, and the organic catalysts that allow these reactions to take place. So you can just draw a map here, H2CO2, pyruvate, amino acids, or 2-oxoacids, amino acids, and so there's nothing surprising here. The pterin cofactors over here, and iron sulfur clusters, and then the standard organic cofactors over here. And then you can count the reactions. It's actually only 402 reactions that are required to get from H2CO2, ammonium phosphate, and H2S, and water to amino acids, bases, and cofactors. You can count them. It's not that difficult, okay? It's a finite process. It's not unimaginable. And furthermore, if you start to look at the delta G of those reactions, green means exargonic, purple means endargonic. These reactions are largely exargonic. How bad is it? It's surprisingly bad. What we see here is that under the conditions of hydrothermal vents, that is non-equilibrium, that means we have more input products than export products, just like metabolism, pH 9 and 80 degrees, 95 to 97% of the reactions in that metabolic core are energy releasing. That means they have a natural tendency to unfold. All right, so, and these are the reactions that we've identified that are responsible for that energy release. That's interesting in its own right. Let's do the movie, and we're almost on time. So, lights down. 5-minute movie, Origin of Life, that summarizes what I've said up until now, and it's an attempt to make this more imaginable. This is the premiere. This is there's still 2 mistakes. The guys just sent it to me last night. Okay, on the early Earth, the moon had already formed. That is, the Earth had already become molten, and it already solidified with all the water raining out on the oceans. And the oceans were twice as deep as they were today, because today there's about an ocean's volume worth of water in magma and the crust, and that in the beginning was on the surface. The surface was a nasty place because of the late heavy bombardment. On the bottom of the ocean, it was nicer. Look at all the water. All right, so here we have hydrothermal vents. These are the black smokers sitting right next to the spreading zones where the water comes in contact with magma. They're much too hot for the origin of life. Lost city type vents, off-ridge vents, 70 to 90 degrees. These are much more interesting for reasons that we will see in just a minute. All right, if we take a look at the geochemical reaction of serpentinization that's going on here, that is shown here. Okay, this is a thing that the guys need to fix, and that's going to cost me another 2,000 euros. They have to put the vents over here, not where they are. These guys do it on purpose. I know. They know I've got money, and so they're playing with me. Over here, we've got the low temperature hydrothermal vent. Here we have the black smokers, where the circulation, the water circulating through the crust and coming up over here is much too hot because it's directly in contact with magma. That's why these vents are much cooler, 70 to 90 degrees when the water comes up, and this sequence needs to be shortened by about 5 seconds because I don't have anything to say here. Yes. Serpentinization, all right? So the water comes in, reacts with iron-2, inexhaustible reserves of iron-2 in the Earth's crust. Generating white balls. This is molecular hydrogen. Sulfur also eluting out of the system, and the oxygen and water being retained in the rocks as magnetite. And then we go to the hydrogen making awaruite, native metals, and this is our cartoon. hydrogen activation. This is actually not entirely wrong. It gets split into hydrides. This is how these compounds actually react on these surfaces. CO2, right? Physisorption and chemisorption, then they start to undergo reactions. And this is why we're seeing what we're seeing in our reactions in large amounts, because these surfaces are catalytically active. Now, it's heterogeneous catalysis. We don't know exactly what the sites are, but they're doing a lot. All right, and so this is a lot of synthesis going on, and that at specific sites in the heterogeneous catalysis, these golden arrows mean energy release. We have exergonic reactions. Metabolism is unfolding from the reaction of H2 and CO2, just like it does in methanogens and acetogens, giving rise to increasingly stable compounds, organic soup, if you will, but continuously synthesized on-site in compartments made of catalytic walls that have a continuous source of energy as long as there's H2 and CO2 interfacing. The products can then, of metabolism can accumulate on the walls, begin to polymerize, giving us more complex organic compounds of any nature, and those can have catalytic ability, giving us a transition into more complex catalysis, autocatalytic networks and feedback, and that gets us to things that we can't explain, but we can draw a picture of it. And this comes into, right, lots of precursors being formed, polymerization into RNA and RNA world. Nobody knows whether this actually happened in early evolution, but everybody assumes it does, so we put it in the movie. And we've got here like RNA recombining and doing all sorts of interesting things, tRNA. And look what happens here. It's the origin of the genetic code. Well, that was fast, but I told you it's a 5-minute movie. Nobody can explain where the origin of the genetic code comes from, neither can we, but we know that it occurred in LUCA, and so we've got it there in compartments that have a continuous source of carbon and energy and nitrogen to supply the process. The origin of DNA, and then the late transition to free-living cells, and its evolution among prokaryotes until the origin of eukaryotes via symbiosis 2 billion years later. So let's go back to the last few slides in the PDF file, please. And those are here. Yes, life is a chemical reaction. From which reaction did it start? An exergonic geochemical reaction, serpentinization. Water. Last slides. This is the E-ring of Saturn. This was actually made by Cassini. The E-ring of Saturn is made of water. It comes from a little moon called Enceladus, and this is a real picture of Enceladus spraying water out of its south pole. That is, and Cassini flew through these plumes. It's water, right? And it contains salts, and it contains a little bit of organic material, which is probably polyaromatic hydrocarbons. And that comes from hydrothermal activity at the South Pole because it's covered with ice, has liquid water, and probably something like serpentinization going on in between. And that's why they think that there's serpentinization-like processes going on. So if this theory is correct, you don't have to have Earth-sized planets. You can have moons around gas planets that could also give rise to this basic chemistry. Water. Auruite, this is also iron nickel, and it has probably created some problems in modern water systems. So, this are some of the people who are doing the work. I won't mention them all. I'll just mention Martina Preiner and Andre Pereira, who've been doing the chemical analyses. And you'll notice that everybody is smiling, and that's because we continue to have very good success publishing papers, and that gives them good jobs. And these are the people who fund our research, the DFG, the German-Israeli program, Volkswagen Stiftung, the Moore-Simons Foundation, and the ERC has been extremely generous to me. Thank you very much to everybody in the audience who had anything to do with that, and thank you to everybody in the audience for your attention. I'll be glad to take a couple minutes of questions. Stayed on time. You don't have to have any questions. You want to use mine? Mine. Any
Hello,
yeah.
Dr. Martin, thank you very much for your very inspiring talk. I have a first question. Since I'm the chairman, I can ask questions when I want. So, when you have your sterilized can of beans and frogs,
yes.
So we put it 4 billion years and nothing will happen.
Right.
How long do I have to wait before your can is going to work?
There are constraints given by geochemists. Okay, so we can say that the zircons, Steve Mochis in Colorado still lays claim to the old evidence for liquid water on Earth at 4.2 billion years, okay? So roughly about 4.2 billion years ago, we had liquid water. Okay, we can't get started before there's liquid water, both for serpentinization, also because water is the solvent of all life compounds, right? All life processes. Then in sediments, 3.8, some say 3.9, but that's more tenuous. 3.8 billion years ago, there is evidence for life in the form of light isotopic carbon. That is carbon that has been fixed by biological processes which discriminate against the heavy isotope. So that gives a window of about 400 million years. Now,
that's all.
Yeah, that's smart. Christian de Duve had a very interesting insight. He said, life is much more likely to evolve in 10,000 years than in 10 billion, because if it takes 10 billion years, it's not going to happen anyway. And I think the limiting principle in this theory is the lifespan of a hydrothermal system.
So did you put away one of your cans so that in 10,000 years we can check if it was true?
Hey, look, I'm 64 years old. I'm
no,
trying to get to age 90, you know, and so
it's clear that it's
I asked the DFG for a funding program that would go for a billion years, and they said no.
Well, you said 10,000 years. Okay. The second thing is these hydrothermal smokers.
Yes.
When I was a director of a company, I spent a lot of money in a professor in Regensburg, and he went with a little submarine, and he had these unmanned submarines. You could go all the way to the black smokers.
Carl.
Yes. Yeah. And a very intriguing man, and
yes.
He took all kinds of samples and published them in Nature too. That you have this bacteria that can live there at plus 100 degrees.
Yes.
And he had this fantastic conclusion that inside the smokers they're very hot, but the ocean is only 2 degrees, so you have a gradient where these bacteria can grow.
Yes.
So then I come to the conclusion that there must also be life in the black smokers.
Actually, the black smokers are full of life. Hyperthermophiles, the record is now 121 degrees. But the most interesting thing is actually, to me, is not so much the smokers themselves. When these drilling programs that go down 3, 4, 5, 6 kilometers, as deep as people go, they can't find anything that's actually sterile. There's always either some dead material or some living cells kilometers deep below the crust. And almost all of it is based on the acetyl-CoA pathway because they have to supply their carbon and their energy needs, and they have very few requirements. They just have to have things that are present there, carbonates and hydrogen. And so these cells are very hungry. And that also provides a rationale for why life didn't evolve many times, because once it evolved, then it spread, circulated, populated the crust, not the surface, the crust, and then they were already there. And so if you come in to occupy a pre-existing, an already occupied niche, it's difficult. And so they were probably capitalizing on all the synthesis that was going on. But that's one of the big surprises.
The black smokers are the same age as the lost cities?
Well, both processes have been going on since there was water on Earth, but the black smokers, because they're on the spreading zones, they move out and then they collapse after about 10 or 50 years because the spreading zone is spreading and they get crushed, and then the veins that are supplying them become crushed, and so they lose their activity and fall down. The off-ridge vents are in much more stable circumstances, and Lost City has an age between 30 and 100,000 years. So that's the kind of time scale that I can imagine. It probably puts a constraint on the time required for the origin of life.
Did you hear of the theory that Mars and Venus lost their water because of this hydrogen producing process, and the hydrogen was too thin, so it was lost in the universe?
Yeah.
And on Earth the water was contained because the hydrogen was captured by life and kept back into the water form. And so that you need life to keep the water and water to get life. So.
Well, okay, so serpentinization has been, okay.
And we are a water institute, we are like a water theorist.
You've got a point, okay, you've got a point. And it's true that the serpentinization is going to continue until there's no more water left on Earth, because there's much more iron 2 on the Earth than there is water. But whether or not, okay, you can make that argument that life kept Earth wet, all right? Okay. I'll let, that's a good argument. Okay. It makes us all feel better.
Any more questions?
Yes, we've got one in the back.
Oh my God, how we get a microphone there? There it's coming. Can you please sit a little bit closer to the front if you want to ask questions this afternoon?
Excuse me.
You have a lot of space here.
Thank you very much for the talk, and I studied the black smoker a little bit actually, so I agree with Case that that can also be a place for the life. But my question is that if you actually have so much organic molecules that can be formed in an abiotic form, so why do they have to go to the autotroph instead of heterotroph? Because like, you know, the black smokers is full of life, yeah, and a lot of them are actually heterotrophic bacteria, yeah, or archaea. Yeah.
So you're asking, where did heterotrophy come from?
Why the life doesn't start with heterotroph, but have to be autotroph?
It's because, all right, so
you just
this is a long, complicated
molecules.
question. This question usually substrate level phosphorylation is the answer. There's only one pathway of substrate level phosphorylation from a biosynthetic process that generates both carbon and energy, and that's the acetate kinase reaction adjunct to the acetyl-CoA pathway. For heterotrophic reactions, if you look at substrate level phosphorylation, it's always oxidation of reduced carbon compounds, always. So the reductive substrate level phosphorylation is the exception. And for these oxidations, you can also say, well, maybe it was, you know, organics from space, but they're too heterogeneous and don't give rise to, then you would have metabolism operating from outwards towards the core rather than from inwards towards the outside, and that would mean that the external reactions would be more conserved than the internal reactions, and the converse is observed. I've got a paper called On the Origin of Heterotrophy, 2016, with Peter Schönheit. It's explained there. Sorry.
Thank you.
Was there already oxygen in the water at the time?
No,
it depends on who you ask. There are some people who say there was always oxygen around coming from, there are water splitting reactions, okay? When you get up to 400 degrees and 400 bars, there's water splitting reactions, okay, but it has no retention time, okay? It's immediately consumed by the reducing
But it's interesting that you say that the organic compounds like, yep, pyruvate and acetate were already there chemically. So
being synthesized,
and oxygen there and sulfate there, so hydrotrophic reactions.
Okay, but get into the,
the
actual physiology of the heterotrophic reactions, and you're into chemiosmosis, and that won't work via substrate level phosphorylation alone. Sorry.
Last question. Anyone? Last question. Dr. Fuchs. Okay.
Well, thank you very much. It was really interesting, and I could follow it, and I completely agree with you. This is a possible origin. But about the organic soup. Now, in your video, the weather was nice. It was sunny. And in these circumstances, I agree that organic soup wouldn't probably do anything. But I've seen many illustrations of the organic soup with a lot of lightning, that being a source of the reactive species that would then form life. Could you please comment on that?
Okay, we can take one of the cans and let it get struck by a bolt of lightning. That will increase the temperature of the contents to about 2,000 degrees in a very short time, and it will end up as a bunch of smoke. We notice that most people who get struck by lightning do not grow faster. They have the lightning is very problematic. We have to get and basically do we have to get a continuous? My argument is that yes, you can increase, you can add energy in the form of lightning, and yes, it's very familiar in representations of the standard heterotrophic theory. But I think what works better as an analogy to the design and function of metabolism is a constant supply of redox chemistry in the form of H2 and CO2. And the organisms that still inhabit the crust today still live from those reactions, so there's continuity. But at the same time, Nobody was there, okay? And it's all hypothesis, and we have to be clear on that there are no answers to these questions, there are only ideas. And furthermore, everybody is entitled to believe exactly what they want to believe on this topic.
Well, thank you very much for your presentation. I must come to the conclusion that we still need a little bit of God to go from this polymerization to the genes, but nevertheless, it's interesting as a water institute to hear that only started when there was water. So I would, now we have a small break, if I'm right. No. Oh, now we go immediately to the parallel sessions, and they are on the screen. And if everything is right, now I forgot, Hester, what session was cancelled now? Was it okay, in the afternoon, one session is cancelled because everybody has COVID. So everybody is informed, but I will talk more about that this afternoon. So I wish you very inspiring parallel sessions. Let's start moving, because we are famous that we're always on time for the lunch. Thank you.